This is All Things Cancer, the podcast of the National Foundation for Cancer Research, hosted by NFCR President and CEO, Dr. Sujuan Ba, a globally recognized cancer research advocate and visionary leader. Each episode explores the hindsight, insight, and foresight of cancer research and care, and how NFCR and its partners are accelerating the path to cures.
Hello, this is Dr. Sujuan Ba, and in this episode, I had the opportunity to facilitate a thoughtful and inspiring conversation between two exceptional scientists who are part of the NFCR network. The first is Dr. Brian Leyland-Jones, a longtime member of NFCR's medical advisor. Dr. Leyland-Jones is a well-known breast cancer physician scientist who has a very impactful career with leadership positions at the National Cancer Institute, McGill University, and the Winship Cancer Center at Emory University. His innovative clinical research work has helped transform how we care for patients around the world. Brian has always been a passionate advocate for advancing science that truly reaches patients. Joining him is Dr. Avery Posey, an incredibly talented and innovative scientist from the Medical School of the University of Pennsylvania. Avery is part of a new generation of researchers pushing the boundaries of immunotherapy. With the funding from NFCR, he's working on innovative CAR-T cell therapies to treat pancreatic cancer and other solid tumors. CAR-T cell therapies have already changed lives of many patients with blood cancers. It was a wonderful and inspiring conversation, one that really shows how collaboration between generations of scientists can accelerate progress in cancer research. I'm so glad we can share it with you here today. Let's take a listen.
So I think it's been about 20 years, and believe it or not, there was a cancer meeting in New York. I think it was in the Marriott. And Webb Cavaney, who I've known for a long time because he was at McGill with me, was in the lobby and introduced me. And since then, it's just built and built and built for me. It's just been a wonderful experience.
So I have a grant that was first an application, now it's funded by NFCR with a collaborating PI. It's been helpful to me to develop a CAR T cell project out of that, which we hope to eventually push into the clinic. So through grant funding. My laboratory is interested in developing immune cell therapies for cancer. And we do this through the development of CAR molecules, chimeric antigen receptors, which are synthetic immune receptors. We are interested in ways to tailor these therapies to be more specific to cancer. And that's different than what has been developed, I think, previously. We try to integrate into our therapeutic development changes in not just protein expression in cancer, but also changes in how that protein is decorated with carbohydrates. We know that tumors change their glycosylation from the normal tissue state to becoming a malignant condition. The change in glycans change, carbohydrates and glycans change. And so we're interested in using antibodies or binders that can recognize that difference. We think that helps us get more specific to tailored therapy for cancer and potentially decrease toxicity. So in this project, we've used an antibody called MAB109, which targets a protein called CCAM6. It's a protein that's of interest in cancer because it's overexpressed in many cancers. But what's special about this antibody is that it has a requirement for a certain type of carbohydrate on CCAM6. And so in the absence of that carbohydrate, it doesn't see it. And we think that helps improve specificity. And then we've modified the receptor further by optimizing the signaling that this type of immune receptor would provide to T cells. to eventually come up with an optimal T cell therapy that is both lytic, it can control tumor growth, but it also has extended survival for the T cells that we think is necessary for combating large masses of tumors. Tumors aren't, as we would see in a cell culture dish, hundreds of thousands or 1 million of tumor cells. There are hundreds of millions of tumor cells, and combating that requires a long time. We need these types of immune cell therapies to persist for a long time.
So why do you think it's taken so much time and energy to make CAR-T therapy work in solid tumors?
So some of the limitations have been identifying appropriate targets. Pursuing targets that tumors can escape has been a severe limitation. We know some of these targets, like mesothelin, is easily escapable by the tumor. If the tumor loses that target, it can continue to survive and then also avoid the therapeutic. This happens in some of the blood-based cancers also with CAR-T cells losing the targeted molecule. In addition, some of those CAR-T cell therapies have caused toxicity, so that can be a limiting event for further pursuit of certain therapeutics. But then solid tumors also have the added complexity of dense tumor microenvironments, immune cell components that are significantly more suppressive and inhibitory to immune cells than in blood-based cancers. And so understanding all of those limitations and the ways in which a tumor can escape these types of therapeutics and then integrating that into the next generation of therapeutics is currently where we are.
So, I mean, you're lucky to be so young, you really are. I mean, so when I graduated from medical school, of course, there was nothing but chemotherapy. And then the first targeted therapist, I think it was approved in 1987, that kind of time. No, it was 97. And then along came immunotherapy. Now we're looking at epigenetics, so many other targets. Where do you think we'll be in 10 years? Where will CAR-T fit in versus checkpoint inhibitors, versus CTLA-4 or LAG-3 or the others?
Yeah, so I think checkpoints definitely will have a longer runway than our current day, maybe 10 to 20 more years of identifying additional checkpoint molecules that can be targeted in combination with anti-CTLA-4, PD-1, and LAG-3. And that may differ by tumor type. It will differ by tumor type. RNA-based therapeutics, as we heard earlier today, are going to be very important in modifying not just RNAs that lead to protein coding, that are protein coding RNAs, but also non-coding. But I'm really interested in carbohydrates as well. So understanding how carbohydrates play a role in protein function and lipid function, and understanding which ones are critical to target is an area that is vastly understudied, mainly because we don't have a lot of researchers in this area, but we also don't have the technologies currently to precisely modulate single residues with a particular glycan. And once we are able to do that, currently we can understand what are the glycans that exist and where do they exist on a protein. But we can't modulate a particular glycan on a particular residue to understand its function. We can modify whole protein level, whole cell level. But once we have that ability, I think it will give us a greater understanding of the complexity of biology in cancer.
Can you do a full glycan map?
Yes.
You can.
That is currently possible. Glycoproteomics has come a long way in the last decade of understanding what carbohydrates exist both in and O, as well as the glycosaminoglycans, where they exist on proteins and what those structures look like. But also genetic editing, CRISPR-based tools, has allowed us to generate cell lines that can produce uniform or homogeneous levels of glycans, and that helps to understand at a cell-based level or a whole protein level what a particular glycan structure might do. But that's not how it naturally exists on cells. There's heterogeneity and complexity there.
So, I've been in the field a long time. So when I started treating patients, as I said, we only have chemotherapy. We basically had no biomarkers, nothing to show us which way to go. And there's a standard cocktail of nasty chemotherapies for pretty much every tumor. Now I sequence pretty much everyone. Can you just help explain to the audience what exactly sequencing is and how important is the DNA and all of the genes and what happens when you get to cancer?
Sure. I mean, cancer is driven by several changes in the cell. Some can be viral, some can be pathogen driven, but most cancers are driven by mutations in those cancers. And so understanding what those mutations are and the functions of those mutations and how it contributes to cancer allows us to develop drugs that can target those particular important mutations to limit tumor growth. And that's likely why you sequence patients' tumors in order to understand what mutations exist. Because
so those are the drivers of the cancer.
To understand the drivers.
And there are several drivers usually.
There are several known drivers, yes. But then also in interest of the immune system's response to cancer, it's interesting to understand what are the mutations that are present and which of those mutations might be presented and shown to the immune system that we could develop immunotherapies to target. Some that may be common amongst patients and some that may be very personalized. And so that becomes very relevant as well.
So, I mean, when you think, I was born in 1949, I say it very easily, and that was 4 years after World War II had ended. And it was the nitrogen mustards that came out of World War II, basically modified toxins, which became the first chemotherapy. Now we've got a whole plethora of different treatment approaches. Yeah. So, I mean, what has been achieved in the last 50 years is utterly, utterly remarkable. You sort of thought, well, there are so many other ways to go, like CRISPR, like microRNAs, like non-coding DNA. And the whole regulatory system for the genome is incredibly complex. I now sequence my patients to give them usually 1 or 2 chemotherapies, usually one, a target therapy plus immunotherapy. And that is how far we've come in 50 years. Sure. Where will we be with CRISPR, CAR-T, non-coding DNAs?
Sure. So CRISPR has given us the ability to not only modify pre-existing mutations that could be useful in preventing development of cancer, but also in the treatment at the regulatory level of the genome, so epigenetics. These are tools that are in development, but they are coming down the pipeline. So within 10 years from now, we'll see drugs in the clinic, at least in clinical testing, that can modify some of those regulatory changes. We heard about a mutation in an H3K27M, a gene that's an epigenetic modifier that's known to be involved in pediatric gliomas today. And that has broad influence on gene expression, those mutations. And using these types of gene-targeted approaches like CRISPR, you can modify those changes back to a normal state, or use them to prevent the changes that are leading to malignancy. So I think that those will come into play. We're starting to see this in other areas of medicine. CRISPR-based therapeutics are under evaluation clinically now.
So, the reason I love NFCR is, well, there's a Harvard saying, which is that culture beats strategy every time. And the culture within the organization is absolutely fantastic. And they're going into strategy and their vision They're going into high-risk research, things that basically normal grant agencies would not fund. They also aim to support novel companies, women in the competitions. They try to work with everyone else. It's not competitive at all. But I think the thing to admire most is the culture, the stamina, the drive of the people to try to bring a close to the cancer. I think there's a phrase from Gladiator, where the chap goes out into the bull ring and he just says, look, whatever comes out of those doors, we're stronger if we bond together instead of, fighting each other. I mean, NFCR is complementary to NCI, to many other organizations, because it specializes in funding that valley of death, getting money in to the big ideas, which are so novel that, no one really appreciates. I remember a friend of mine was given an award at AACR for a particular technique, won't go into details, and for the first 10 minutes, he showed his 10 grant refusals to his grant applications. This will never go anywhere. This is just rubbish. I don't know what these people are talking about. And then 10 years later, he got the award for it. That is what NFCR funds. It's that brilliant people, some older, some younger like yourself, but they fund that like impossible dream. And that's what makes NFCR so special. So why should it compete with anybody? It's complementary to everybody.
Sure.
It has an emphasis on, highly novel technologies.
Sure.
And it's, as I said, culture is everything. The people in it are remarkable, faithful, they love each other, there's no backbiting, anything. It's all towards the cure.
And I think you mentioned that most grant funding happens after the work has already been done. This is
Yes,
true because
yes,
the long process
yes.
for review at NIH and other organizations, that by the time you've submitted, it's been reviewed, you resubmit, it's been reviewed again, and then you get the grant, you've already completed majority of the work, if not all of it.
You only write a grant that you've already performed.
Yeah.
That's basically what it is.
So for an early stage investigator, that's really difficult.
Of course.
When you have limited funds to run your operation, to do work that is unfunded with the hope of it eventually being funded at some point is it's tough.
So did you partner with anybody else on the NFCR funding?
I did.
I thought you said
I did,
you
yeah. So I have a clinical collaborator. He's at the University of Oklahoma Health Sciences Center, and he's a gastroenterologist, and he studies the types of diseases that we're hoping to treat. With his help, we've been able to test our CAR-T cell therapies, not only in cell cultures with cell lines and in our xenograft models with tumor cell lines, but also in patient-derived organoids and in patient-derived xenografts. So we know that this has direct relevance to patient samples, and I think that helps us get closer to the clinic than would be without that collaboration.
Strongly agree. I mean, you have to encourage novel ideas, young investigators, and also, some of the older people have brilliant ideas in their 60s and 70s because, they walk the walk. They often say that only 1 out of 10 ideas makes it, through to the end. But if you fund 100 of them, then that's 10 that are coming through and can move onwards. Can I flip onto something which is more personal? Prevention and early detection. Where do you think we will be on that? Charlie Swanton published a famous paper in Nature Medicine.
TraceRx study.
Well, yes, pre-TraceRx, he published a famous paper which said that just on basically size, you could not pick up a tumor in liquid biopsy until it had reached 1 centimeter. By the time it reaches 1 centimeter, there are 100 million cancer cells in there. So how do we detect early, and what do you think the treatment strategies will be early?
Well, from that information, it's increasing the sensitivity of the biopsies. Some of the work that is being done by several of the companies that are doing this and the academic groups, I think are incorporating not only cell-free DNA, circulating tumor cells, but also what's encoded in exosomes, which has become an interesting area because those can contain proteins associated with the tumor, RNAs associated with the tumor, and they contribute to the oncogenesis of the tumor, along with the other macromolecules, carbohydrates as well. Carbohydrates are used in the detection of cancer as well, like CA19-9, CA125, et cetera. So I think increasing the sensitivity of these tests, I'm encouraged by some of the work that's come out of these companies that are working on these blood-based tests to detect cancer. I think there were studies that had come out in previous years that could show the ability to detect cancer, not only that a patient has cancer, but the anatomical location of that cancer based on the molecules that they're identifying.
Yeah, based upon the changed genes.
Correct.
The microenvironment has to be totally in place before the tumor forms. So what do you think of measuring proteins which are shed from the tumor microenvironment into the blood?
Very interesting. I think that the stromal cells within the tumor are incredibly interesting. The changes that happens to the stromal cells and how they support the niche for a tumor to grow, how they support new niches for metastases to grow. I think there's evidence that cancer-associated fibroblasts establish new niches in a metastatic site before tumor cells arrive.
Yep.
And that, I think, points to the importance of studying the cell type and understanding the changes that are occurring in those cell types. And not only the cells themselves, but the matrices that they are secreting and establishing. My lab is interested in this. We are pursuing a type of extracellular matrix protein called fibronectin. Fibronectin is known to have many changes in the isoforms of fibronectin that exist in cancer, but also in the post-translational modifications that exist. We have CAR-T cells that can target a version of fibronectin that we think is cancer-specific, and at least in mice, can control tumor growth. And we think that this is an area that is ripe for investigation. The tumor stroma, as well as the extracellular matrix, and the immune cells that are part of that, that may be contributors, maybe initiators. I think it's important areas of investigation.
I mean, I'm sure that you're aware of this, but if you're measuring, say, like 6 tumor-related, TME, tumor microenvironment proteins, if you're just looking at, say, 6 of those in the blood, they can be run in a standard clinical chemistry assay. And some of my colleagues can run that like for $10 or $20 because it just goes straight into the automated robotic, CBC machine. So that would be huge because it's early detection and it's incredibly cheap.
Yeah, I mean, that may mean that in addition to looking for particular mutations that we know are common in cancer, there are mutations that are the drivers of cancer, looking at proteins or protein coding or non-coding information that may be more specific to the stromal cells that have made these alterations.
When do you think we'll make a big dent on early detection prevention? I mean, it's been going for many, many years. I'm sure that, most of our patients are asking why on earth we haven't reached the point where we can do this.
I think in some areas this does exist. I mean, I'm not a clinician, but I think that the at-home tests for colorectal cancer points us in that direction, that it's possible with certain cancers to be able to detect to do early diagnosis. Maybe more challenging for others, but I think there are ways around that, hopefully within the next 10 years.
So sequencing of the cancer genome.
Of the cancer genome or the human genome?
the cancer. The ability, like for foundation medicine, as opposed to doing the complete.
Yeah.
But the panels, I believe, came out in 2007.
Okay.
So when we get a patient in the hospital, the very first time we see them, we walk them through their prognosis, how we're going to treat it, and we sequence them at diagnosis. And then you can put together, if you've got, say, 2 targets in there, you can usually guess which is the primary driver, and we can use a targeted therapy for that. Then we can use a baseline chemotherapy or baseline radiation, both of which stimulate the immune system, which is very important. And then we'll add an immune therapy on as well. So with that, we've taken on our we did a trial called iPredict, which was published in Nature Medicine. And in that, we showed a hazard ratio of 0.4, which means a 60% improvement in overall survival. In a randomized trial, when we did sequencing and gave targeted and immunotherapies versus state-of-the-art NCCN guidelines. And I think just how much we have achieved. It's been remarkable in especially the last 25 years. But you also become aware of just how much we don't know.
Yeah.
But the encouraging thing to me, Avery, is that the patients are now benefiting from at least the sequencing, the targeted therapies, the immunotherapies. We showed it's 60% better if you target, sequence, give immunotherapy, and who knows? So we've already got a great baseline, and now it's a matter of just taking it up, just incrementally, looking at the next generation of companies. It gives me the vista to see the entire scientific community.
Yeah, so I talked to entrepreneurs who are developing therapeutics, as well as supporting the development of therapeutics, including some that have large facilities with over 5,000 employees, developing CAR-T cell therapies, supporting 30 plus investigator-initiated trials. And I think that collaboration with groups like that really helps to push forward science, helps push forward innovation, and get us to answering questions of which modalities have the best efficacy, which ones should be moving forward, and which ones should not. I think that this is really important with such a plethora of modalities and potential therapeutics to use. I think it's important to collaborate in order to accomplish those goals.
Thank you for listening to this episode of All Things Cancer. Be sure to subscribe so you never miss future conversations. To learn more about how the National Foundation for Cancer Research is working with the global research community to make cures possible, visit NFCR.org. You can also reach us directly at 800-321-CURE.